Okay everyone, today we're going to learn about meiosis. By the end of this video, you should be able to identify the phases of meiosis and arrange each step in the correct sequence. So I'm going to explain what each phase looks like and what sequence they go in. First, it's important to remember what we discussed when we discussed mitosis. Okay. When we discussed mitosis, we said that you take one cell and it's going to divide and make two genetically identical cells. So the two cells that it makes are identical to the original cell. And the cells that mitosis makes are body cells. That would be things like skin cells and muscle cells. Those would be examples of body cells. Contrast that with meiosis. So in meiosis, you make genetically different, not identical cells. They should be genetically different cells. These cells that get made are sex cells. So that would be things like sperm and eggs. Specifically, sex cells get made in meiosis. Okay, meiosis only makes the sex cells, sperm and eggs. Now, mitosis, go back to it. It goes through PMAT. Remember, PMAT's prophase, metaphase, anaphase, telophase. So when the cell divides, it has to go through those phases. Meiosis does PMAT as well, but the difference is mitosis is only going to go through one cycle of PMAT, one round of division, but meiosis is going to go through two rounds of that, 2 rounds of PMAT or 2 divisions. And you can see this in the picture that I've drawn. Here's our original cell, and it divides only one time. And so you get 2 cells. But when you do meiosis, there are 2 rounds of division. So we take our original cell, we divide it the first time, and that's going to give us 2 cells. So far that's like mitosis, but the difference is now each of those are going to divide. And so by the end of meiosis, you have 4 cells total. 1, 2, 3, 4. And it's important to know that the 4 cells that you make in meiosis are what we call haploid. On the other hand, the 2 cells that we make in mitosis are diploid. So what's the difference? Well, diploid just means that you have 2 copies of each chromosome in that cell. One of the copies is going to come from dad, and the other copy is going to come from mom. Meiosis, on the other hand, is only going to have 1 copy of each chromosome. That's what we mean by haploid. 1 copy of each chromosome. So, you in a sperm, if this is a sperm cell, it will have only 1 copy for each chromosome in it. That copy may be from dad, that copy may be from mom, it may be some mixture of the two. So we're going to see how this works. Just to kind of illustrate this in a nicer, neater picture, this blue chromosome here, we're saying that comes from dad. And this red chromosome here, that comes from mom. Now you'll notice that the 2 chromosomes here are similar in size and shape. So they're roughly the same height, they're roughly the same shape and everything. So we would call this, because they're similar in size and shape, we would call this a homologous pair of chromosomes. So this right here is a homologous pair of chromosomes. Now, after the DNA gets replicated, you're still going to have a homologous pair, but now it looks like you have doubled the material there. So it's important to remember that after DNA replication, this would be a sister chromatid. And this is a sister chromatid as well. So, in this picture here, how many chromatids are there in total? Well, it looks like there are 2 sister chromatids from dad, and there's 2 sister chromatids from mom. So that would be 4 sister chromatids total. Now that's going to be important to know the difference between a chromosome and a chromatid later, and what is a homologous pair and what isn't. Alright, so let's go through the phases of meiosis here. We start out in before meiosis begins. You start out in interphase. So this is not one of the phases of meiosis, it's leading up to it. And we have, if you look inside this cell, 4 chromosomes to begin with. 1, 2, 3, 4. Now, we're going to go through DNA replication. Alright, so the DNA is going to unwind. DNA replication. The DNA is going to unwind. That's what you see going on here. And all of it's going to get copied. And so, by the end of interphase, now we've doubled the number of chromatids that we have. That's why it looks like there's double all the material. We still have 4 chromosomes total, but now we have 8 chromatids. And then this is where meiosis is going to begin. So we start, remember our PMAT, P-M-A-T. We start in prophase. And what's going to happen is homologous pairs of chromosomes, that's the chromosomes that are similar in size and shape, are going to pair up together. And when they pair up together, you're going to see some, what we call crossing over. And that's where these little bands swap places between dad's blue chromosome and mom's red chromosome. And so that means some of dad's genes get onto swap places with some of mom's genes. And then once they swap places, now you have this unique combination of genes that you didn't have before. Then it goes into metaphase. That cell goes into metaphase. And remember, in metaphase, we say M is for middle because the chromosomes line up in the middle. So what is it that's lining up? It's homologous pairs. This is a homologous pair, and this is a homologous pair. They're similar in size, they're similar in shape. So their homologous pairs line up in the center of the cell, that's metaphase. And then during anaphase, the homologous pair gets separated. We say A is for away, anaphase starts with an A, so the homologous pairs move away from each other, they get separated. And they move to the sides of the cell. And then in telophase, the cell is beginning to divide. it looks like you're about to get 2 new cells. T is for 2, we say, for telophase. And cytokinesis is actually the step where you actually do cut the cell in half. So now, notice, we started out with 4 chromosomes in the cell total, but by the end here, we've got only 2, only half that number, because we started out with 2 chromosomes from dad and 2 chromosomes from mom. Now, each cell is only going to have 1 copy of each chromosome, not 2 copies of each. So that was the first round of division. Now we're going to go through the second round of division. So this is round 2. Okay, so you'll see these little 2s here just to indicate that's the second round of division. So you're going to go through the same process though. You have 2 new cells here in prophase 2, and those chromosomes are going to move to the center of the cell in metaphase. M is for middle. The spindle fibers attached. That's these little, they look like strings, and then those spindle fibers are going to pull the chromatids apart. So in anaphase, it is chromatids that are getting separated. And then finally, in telophase 2, you have the cell, each of these cells is going to divide. And so in telophase 2, you get 4 cells. And if you look at what's inside of them, each one of those cells is different. Here I have a long blue chromosome with a small red portion, but here I've got a long red chromosome with a small blue portion. So that's what makes each sperm cell and each egg cell different. It's a unique combination of genes, and it's when you bring that sperm and egg cell together that you're going to get a unique combination, and that's why children can look very different from one another, even though they come from the same parent, because during this step called crossing over, we got a very unique combination of genes that swapped places. All right, that's all.